to the first seminar this year, right? And welcome the chair, and welcome all the students, the faculty. Happy New Year, belatedly, to all of you. This seminar is sort of typical for our midwinter seminars, as we invite faculty and students from the area here because of the weather. You see, today is not the most friendliest weather, but it came. So now Emma Teal is a special student, and I'm delighted. This is a new experiment for me at the end of a career, that we invite a PhD student to a major seminar in our department. It's interesting, I just was talking with her, the American Physiological Society, to which she belongs, has more and more incorporated into their committees, its committees, students, graduate students, and playing more and more important role. That's where we bring the youth in. Now, having said that, this is a special student from my point of view, and I must tell you, when I was in Emma's age, 60 plus years ago, we were rebuilding Germany, and we were out in such a weather to find the bricks to rebuild the Department of Anatomy. I'll never forget this after World War II. So we didn't have, although we were had the initiative to study, but we couldn't study because we had to rebuild our buildings and our society basically. So, so here is now Emma. She will talk today about a very important topic: metaplasia and tissue repair in gastric injury. Let me tell you a few things about her. Between 2010 and 2015, she was undergraduate student at the Department of Biological Sciences in Cincinnati, and she had a BSc in the Department of Mathematical Sciences. That's what we want in physiology, because physiology is a quantitative science, thinkers. Then the capstone title she had at that time was Mathematically Modeling Seed Harvesting Techniques for American Chestnut Restoration Populations to Conserve Genetic Diversity. Look, she worked on chestnut genetics basically. Look how different it is from what you are doing here and what she's doing now. But that's the whole thing. The investigative mind doesn't know boundaries and wants to look at everything. Okay. This morning, my wife and I, we drove in. Our driver is here, and she's part of this invitation, and we discussed what is the cause. How could climate change the ratio of boys to girls, which has been found in Japan now? That's an interesting topic. So we could delve into it and study it and explore it. Now, from 2015 on, after that, she became a graduate student, PhD student, in systems biology and physiology, molecular and cellular physiology. A thesis, which is finishing this year, the role of CD44 variant 9 in gastric ulcer repair. That you were going to hear about this. So she had received 6 honors. One of them was the Ohio Physiological Travel Award. And that travel award pays enough for you to go to the East or West Coast. And she showed up at San Diego, and that's where I met her. With Norma together, we were at an APS dinner, and I think the entire dinner talking with you about the question, what makes a normal cell a cancer cell? And the question, how do we explain the famous Warburg effect? And Warburg was a German Nobel laureate, who was 87 when I had lunch with him in Freiburg, in Germany. So there's a long line of history. She has, in presentations, she has more than a dozen presentations in the area. Some of them go way back to the chestnut story, but now many of them are in the area where she's now working. She has publications. She lists 4 publications, and 2 are in print. Okay. So one of the other aspects of her undergraduate studies, pre-graduate studies, was that she didn't stop with chestnuts to study genetics of chestnuts. She studied music. She studied Spanish, and she studied composition theory, right? That's what she did. So look at that. It reminded me of my days when I started studying physiology. I was studying at the same time parallel music at the University of Freiburg. So this is a talent that does not know the boundaries, and that's what I think you will see in her talk, and that's so inspiring for all of you. That's why I invited her. Emma, it is all yours now. We welcome you. Okay, you need a cookie?
Sure. Thank you. Okay. Can you guys hear me okay? Does this mic work? Okay. Okay. So I was just thinking about it before I start. The last time I was here was a decade ago for band camp in high school. I've never actually been here before, so it was kind of funny. It was almost exactly a decade ago. So, but anyways, thank you for the wonderful introduction, Dr. Lauf. My name's Emma, and the title of my talk is Metaplasia and Tissue Repair and Gastric Injury. So, as you all know, or I hope you know, the stomach serves to initiate the digestion of food. That's really its primary purpose. And in particular, the fundic region secretes acid to help do this. It digests food, but also, importantly, it also serves to protect the stomach and really the body against any foreign pathogens that might be trying to infiltrate. So the fundic glands contain a unique composition of specialized cells, and these include our acid-secreting, mucus-secreting, zymogen-secreting cells, and our histamine-secreting cells. And additionally, the antral glands also have their own specialized cells. Now, a gastric ulcer really is, let me define it for you, it's a lesion within the mucosa of the stomach and extends all the way down to the muscle layer of the stomach. And in some cases, if it's bad enough, it can even perforate the stomach. And over 5 million individuals are affected by gastric ulcers. So this is really something that's affecting a lot of people. And we have over 500,000 new cases of gastric ulcers each year. Now, the primary culprits for gastric ulcers are Helicobacter pylori infection, which is a gram-negative bacteria, and also non-steroidal anti-inflammatory drugs, or NSAIDs, like Aleve. So these are the leading culprits for gastric ulcer disease. Now, gastric ulcer disease, or gastric wound repair, is very important, and the reason why it's so important is, as I mentioned earlier, the stomach is one of the first lines of defenses against any pathogens that might be trying to infiltrate the body. So it's really critical for the stomach to quickly and efficiently re-epithelialize any wounds that it might have. And in addition, it's not only important for it to re-epithelialize, but it's also really important for it to really gain back that function that was lost. So it wants to recreate those glands that were lost. So to do this, during repair, stem cells will migrate from the base of the ulcer to the epithelium to form the new gastric glands. And during regeneration in the intestine, epithelial stem cells from the base of the ulcer margin will form these tubes that are composed of what are called ulcer-associated cell lineages, or UACLs, because it's kind of a mouthful. So what you can see here, this is actually a mouse stomach with an ulcer. So you have the ulcer here, you have the ulcer margin on either side. So you'll have stem cells migrating to re-epithelialize the wound area. And here you can see these are some nice cross sections of some UACLs. So these are our ulcer-associated cell lineages, that will form our new gastric glands with all of these specialized cells. So it's a very complicated and fascinating process. There's a lot going on here. Now, one phenomenon that is observed during gastric repair is what is called SPEM, right? It's short for spasmolytic polypeptide expressing metaplasia. And traditionally, SPEM has been studied in cancer, right? However, in the setting of parietal cell atrophy, chief cells will reprogram, transdifferentiate into cells that are driving metaplasia. So these are really very metaplastic cells, and we see it during gastric wound repair as well. So these chief cells will be converted into these metaplastic cells that are highly expressing TFF2, which is a spasmolytic polypeptide, hence the name spasmolytic polypeptide TFF2 expressing metaplasia. In addition, recently CD44 variant 9 has also been identified as a marker of SPEM. It's a very robust marker of SPEM, and our lab has been very involved with really identifying this as a really robust SPEM marker. Now, this is, I'm going to give you guys a little bit of a background because I'm assuming there aren't a lot of people here that are studying gastric repair. So this is a paper that has come out of our lab, and what you're looking at here is intrinsic factor and GS2 lectin, which are really, when being expressed together, is a very robust marker of SPEM. And so you can see these are cross-sections of mice with ulcers, 1, 3, 5, and 7 days post-ulceration. And you can see that we have a nice co-expression of these markers after repair, really demonstrating that, that SPEM may be involved in the gastric repair process. Now, these are transient as well, which means that they're turning on when the ulcer is being initiated, and then they turn off after the cessation of repair. And to really drive this home even more, as I said, CD44 variant 9 is also a very robust marker. And you can see that CD44 variant 9 is very robustly expressed in the stomach during repair. This is at the ulcer margin of a gastric ulcer in mice. Now, what exactly is CD44? So CD44 stands for cluster of differentiation gene 44, and it has been shown to coordinate both normal and metaplastic epithelial cell proliferation. So CD44-9 in particular, there's 12 different isoforms, the standard being a hematopoietic stem cell marker. But CD44 variant 9 is a marker for gastric cancer stem cells, and it also emerges, as I showed you earlier, during regeneration and repair of the gastric epithelium after injury and at the gastric ulcer margin, where you see all of those stem cells coming in to re-epithelialize the wound. Now, importantly, we've demonstrated in our lab that CD44 variant 9 contributes to the gastric epithelial regeneration in response to injury, and it has been identified as a marker of SPEM. So I really want to make this, I want to really drive this point home that CD44 variant 9 is really involved in the gastric repair process. So up until this point, we've shown that it turns on during repair and it turns off, but we haven't really shown that it is actually contributing anything. So Nina Bertoskyrik from our lab took this really interesting approach to demonstrate that CD44 is actually important and very much necessary for gastric cancer or gastric repair. So she took this very unique approach. I think it's really cool. What she did was she gave mice ulcers, right? And she could do this with acetic acid. She could take the stomach out, anesthetize the mice, externalize the stomach, apply acetic acid, and then re-internalize the stomach, suture them up, and send them on their merry way. Now, what she did was she took gastric organoids, She created gastric organoids from black 6 mice and mice that did not express CD44. And she orthotopically transplanted these at the injury site. So she had CD44 knockout mice that she gave ulcers to, black 6, and then CD44 knockout mice that were transplanted with black 6 and 44 knockout organoids. And what she found was really, really striking. So these are our CD44 knockout mice. And you can see this is expression of variant 9 and BRDU for proliferation. And you can see after 1, 3, 5, and 7, we have no expression of CD44, right? This is to be expected. Similarly, when she took black 6 mice, you can see this nice emergence of CD44 variant 9. We have this nice proliferation here at the ulcer margin, and it continues through for day 5 and 7, right? So we have this emergence of variant 9, this proliferation to heal. However, interestingly, was when she orthotopically transplanted black 6 derived organoids into the CD44 knockout mice, you can see we have this emergence of variant 9 and BRDU at the ulcer margin, right? So this is really successfully demonstrating a successful orthotopic transplantation of black 6 organoids into the epithelial tissue of these mice. Now, what she then did was she took measurements of the ulcer size of these mice. And what you can see is we had persistent, in the CD44 knockout and in the CD44 knockout transplanted with 44 knockout organoids, we had persistent ulcers, right? They didn't heal. However, in the black 6 and the 44 knockout transplanted with black 6 organoids, we had very similar resolution of these injuries almost 3 days after. So showing here that CD44 is indeed important for resolution of these injuries. So that was Nina's thesis project in a nutshell. And now I came in, and what I've really been interested in is trying to understand, okay, so CD44 variant 9 is important for gastric repair and for ulcer resolution, but I wanted to know what exactly is it doing. And so I came into this lab reading a lot about CD44 variant 9 and trying to see what what functions are known about it. And what I found was that that CD44 can actually act as a sort of, it actually has this really interesting interaction with what is called the XCT transporter. It's a cysteine glutamate transporter. It's stabilized by CD44 variant 9, and it potentiates defense against reactive oxygen species. So after injury, there's a normal buildup of reactive oxygen species at the injury site. So this suggests that there should be, or you would expect to see, some sort of mechanism to protect the stomach against this buildup. Because at high levels, it really is detrimental to wound repair. It's not a very conducive environment for repair. So XCT can provide protection by cysteine uptake, glutathione synthesis, and maintenance of the redox balance within the intracellular environment. And I put a nice figure here so you can understand this a little more, because it's kind of complicated. So as I mentioned, ROS has traditionally been considered as toxic, and it does have potential, and the reason for this is it has potential to damage lipids, proteins, and even DNA, as well as initiate pro-apoptotic pathways. But it does have some good aspects to it. So it can defend against infection, and it also can act as a second messenger during wound healing. However, as I said, at levels exceeding physiologically normal, it can be detrimental to wound repair. So the stomach does need a mechanism by which it can protect itself. So there has been a lot of literature behind XCT and ROS. And so with this paper here, I won't delve into the details, but this paper here demonstrates that with XCT loss, you have a decrease in metastatic potential, a decrease in metastatic potential, as well as cell cycle, cell growth. So this paper is looking at gastric and as well as lung cancer. And so here what they've shown is in a drug that I'll be showing you that I used is sulfasalazine. It's a very potent XCT inhibitor. And what they showed is with XCT, drug inhibition by sulfasalazine, they see a decrease in metastasis and a decrease in cell cycle, which are both processes that are fundamental for wound repair. So the mechanism that they described in this paper and that I've also demonstrated here is they found that XCT can bring cysteine in, which is the rate-limiting reagent for glutathione synthesis, and glutathione can really inhibit ROS, right? And so what I've done is I've inhibited XCT pharmacologically with sulfasalazine, which there will kind of block this downstream pathway, and so you have buildup of reactive oxygen species. Now, reactive oxygen species have been shown to induce p38 MAP kinase signaling, which leads to an upregulation in caveolin-1. And caveolin-1 is this really interesting interaction with beta-catenin, where it can redirect it from the nucleus to the cell membrane, where it can link up with beta-catenin and E-cadherin to have these downstream effects of decreased cell proliferation and increased cell-cell adherence and decreased cell migration. So again, these are properties that are very important for gastric wound repair and that I'll be demonstrating here. So trying to show this, the first thing that I did was I came in and I ordered these gastric ulcer arrays, and I stained these for CD44 variant 9 and XCT. So these are patients that have gastric ulcers, and you can see this increase in expression in CD44 variant 9 at the ulcer margin in these people's at the margin of these people's gastric ulcers, which suggests that it might be there's a reason might be turned on, right? So and you might also be seeing everything that I showed you earlier was in mice, but we also may be seeing this in humans as well. So the first thing that I did was I used the same mouse model that I showed you earlier. This is a mouse stomach, so I can externalize the mouse stomach. I can apply acetic acid for about 30 seconds. I can re-internalize the stomach, suture them up, and then I can treat them with either a vehicle, sulfasalazine, SSZ, which is our XCT inhibitor, or a glutathione inhibitor, that's the GSH. So I treated them with these for 1, 3, 5, 7, up to 30 days, and I sacked them at these time points. Now, what was really striking was in these mice that were treated with sulfasalazine, we had this continuous, persistent ulcers, right? However, in the vehicle, we had resolution of these ulcers, suggesting, again, that XCT might be important for the resolution of these injuries. And I also quantified these, and you can see that while the sulfasalazine healed after about 30 days, well, not even 30 days, maybe a little bit after, but they really persisted for almost a month, which is really contrary to what you see compared to the vehicle, right? Now, I took sections of these mice. This is 3, 5, 7, 14, and 30 days after, and you can see that in the vehicle treated, we have, again, this transient expression of variant 9 and XCT. However, in the sulfasalazine, what was really interesting, you can see the ulcers here, they didn't really, they just kind of persisted. In the sulfasalazine, it was kind of not what we expected. We saw that CD44 variant 9 with no expression of XCT, but we saw that CD44 variant 9 just kind of persisted even out to 30 days, which is really cool. And you can see here, this is a day 7 mouse section. You can see in the yellow here, this is the CD44 variant 9 and XCT co-expression. So I showed you the sulfasalazine treated, the vehicle resolution, the wounds healed. And in the sulfasalazine, they healed, but they just kind of persisted for a long time, right? But what I didn't tell you was that when I looked at these ulcers, it almost looked they had some kind of cancer development. They were just thick, a lot of fibrosis. We took sections of these, and you can see with an H&E staining that they definitely had some sort of, they looked almost hyperplastic, right? So what I'm trying to say is they healed, but it was not normal healing, right? So next what I did was I thought, well, we have this thickening up the epithelium. What is going on here? We must have some abnormally high, cell proliferation, right? However, when I took this and when I took these sections and I stained them for BRDU and I looked at proliferation, we actually saw a decrease in a decrease in the sulfasalazine treated group. So we actually didn't have any more proliferation than what you would expect. So I treated these for trichrome stain, and what we found was we actually saw just massive fibrosis, right? So the thickening was actually due to this increased fibrotic tissue in the sulfasalazine treated mice, which is, this was very encouraging to us because ROS there's such a strong backing of a lot of really good research showing that ROS is linked with fibrotic development. So we've demonstrated here with XCT inhibition that there is, with XCT inhibition by sulfasalazine, we have a loss and repair in this fibrotic development and abnormal healing. But I really wanted to delve more into the mechanism. Is this mechanism that has been shown in the literature in all these other systems being shown in the stomach as well? So what I've taken here is I've performed Western blots for caveolin, which is down here, as well as the activated form of beta-catenin. So this is the transcriptionally activated beta-catenin. And I got this antibody from a researcher down at NKU who specifically studies beta-catenin. So it has a serine 552 residue that's phosphorylated on the serine 552 residue that shows that it's transcriptionally activated. And what we found in the sulfasalazine group, we had this increase in caveolin, which is to be expected. And similarly, we found a decrease in transcriptionally activated beta-catenin, right? So we have a redirection of beta-catenin away from the nucleus where it can be transcriptionally activated into the membrane, right? So we have that linking that I talked about earlier. Oh, here it is. So we have this increase in cell-cell adherence because it's being moved to the membrane, and we have a decrease in any transcriptional activation by beta-catenin.
I have a question there.
Yeah.
Is the caveolin, that's quite an increase in caveolin.
Yeah.
So that raises the question, you're probably aware of that the sodium potassium ADP is scaffolded by caveolin.
Mm-hmm.
Does anybody look into that? And the enzyme isoform apparently switches in cancer, so would be.
Yeah, that would be interesting. Not that I know of, nobody's looked at the sodium potassium pump with caveolin, but that would be an interesting question because in the end, this is a drug that they do treat a lot of people with this. This is approved by the FDA. So it would be an interesting thing to look at for that sake, but it's not something we've looked at. That's a good point. Another phenomenon that's another process that's very important for gastric repair is what is called epithelial to mesenchymal transition, or EMT. And something that I've been really interested in is trying to see if, and this is something that's never actually been shown in the stomach, actually, is if EMT is actually occurring in the stomach. So that was something that I was really interested in. And what I did was I injured mice. I gave them acetic acid-induced injuries, as I showed you before. And then I FACS sorted CD44 variant 9 positive cells from the injury site to see if these cells are expressing EMT markers. So this is just to show you kind of where the variant 9 expressing cells are. So I FACS sorted these from, I induced an injury in the fundus, and I collected cells from both the antrum and the fundus, with the antrum being our control, and our uninjured control. And so I fact sorted these for gastric epithelial cells and then further for CD44 variant 9. And what was really astounding was this massive, these are all markers for EMT. We found this massive increase in CD44 variant 9 positive cells in EMT markers, and then this lack of expression in the CD44 variant 9 positive cells in the uninjured antrum. So this was really interesting to us, and nobody's demonstrated it before. So I've showed you the mouse work that I've worked on, but really something that our lab really specializes in are gastric organoids. I don't know if there's a lot of gastric or if there's any organoid work at Wright State, from what I've heard. But what we can do is we have this really nice collaboration with a few surgeons at Cincinnati Children's, and they do perform sleeve gastrectomies, and they'll basically take that tissue from these people and just give it to me. Right, so if you've ever wanted to see a human stomach, this is it here. And what I can do is I can basically take the epithelium from this stomach, digest it in this BSA and collagenase mixture, and extract the stomach glands, which contain our resident stem cells. And I can take these and embed them in in Matrigel, which is kind of, it's this gel that kind of simulates the basement membrane of the stomach. And what will happen is the resident stem cells in these glands, we can throw a bunch of growth factors at it, and they will proliferate and grow up into these three-dimensional organoids. Now, what's really unique about these three-dimensional organoids and what makes them more favorable than your normal cell culture is that they express all of these different cell types that you would see in a stomach. So they have parietal cells, they have mucus-secreting cells, they have all of these different cells that you see in the stomach. Now, one method that I've really pioneered and published in Jove and MIMB is this method where I can take these organoids and put them down onto collagen or Matrigel-coated plates where they'll flatten out to form kind of like these little organoid pancakes, and they'll proliferate and create these monolayers. And I call them human gastric epithelial monolayers, and they still express all of these different cell types, as I'll show you. And you can take these and you can scratch them and perform scratch wound assays, which are kind of, they're really a standard in the field of regeneration. You can study cell proliferation and cell migration using these assays. So this is an example. This is my monolayer. We can scratch it, just using a pipette tip or a razor blade, and we can watch them as they migrate and really re-epithelialize the plate. So this is a nice picture that I took, an immunofluorescence picture I took of these monolayers. The red is the E-cadherin, blue being our nuclear stain, and then UEA1, which is a mucus cell marker. And the reason why I'm showing you the mucus cell marker is because you can see I took a Z stack, and you can see that we have a polarized monolayer. So we have the mucus being secreted on one side, so we have a basolateral and an apical side to these cultures. I also performed PCR to look at these and compared them to primary tissue, primary stomach glands that we digested down, 3D human fundic organoids, and then my gastric epithelial monolayers on the far right. And you can see they all are expressing all of these different cells similarly. So I scratched these, and I treated them with sulfasalazine and vehicle. So I'll show you the vehicle first. And I took a time course. They're a little quick, but you can see I scratched, and we have this nice resolution of injury after about 24 hours, right? However, when I treated these with sulfasalazine, you can see we have this loss in cell migration, You can see it almost looks like they're trying to reach out and heal, but they can't, right? And I've done this dozens of times, and we have the same situation every time. This is just one of them. So we see almost no healing in these cultures. So this was really exciting. It was really cool. And it really is demonstrating that XCT is really necessary for our, to show what's happening. We see this loss and repair. So we quantified these, and what I didn't show you, we also treated these with glutathione inhibitor. And compared to the vehicle, which is in the white, we saw this loss and repair in the sulfasalazine and the glutathione. So suggesting, again, that this process, this pathway that I showed you, is really necessary for repair. So next what I did was I scratched these and I stained them for XCT and CD44 variant 9, and you saw this, you can see a nice increase and really beautiful expression of CD44 variant 9 and XCT, a co-expression at, I guess you could say, the wound margin or the wound edge. This is 16 hours after the scratch. And again, I performed Western blots on these cultures, 0, 8, 16, and 24 hours after scratch. And you can see we had an increase in caveolin. We had a decrease in the activated form of beta-catenin. And similarly, I performed a Ross stain on these cultures, and you can see, compared to the vehicle, we really had this tremendous increase in reactive oxygen species in the sulfasalazine and the glutathione inhibitor. So this is really important because gastric ulcers really affect a lot of people in the United States, and the complications following injury are a burden that they predominantly affect the elderly population. So I really want to focus on this. I wanted to look at this in a translational aspect, right? So next, what I'll be showing you is how this affects the elderly population. So it's been established that the incidence of gastric ulcers in the elderly is primarily due to increased use of NSAIDs, as well as an increased incidence of Helicobacter pylori infection in the aged individual. Now it's kind of a double whammy because while they have this increased use of NSAIDs, increased Helicobacter pylori infection, they also have a decreased capacity to repair, right? So they'll get these ulcers and they just can't repair them. So next what I did was I took this gastric ulcer array with both young and aged individuals, and I stained them for CD44 variant 9 and XCT. And you can see in the young, I don't think the projector is showing it very well, but we had this huge increase in variant 9 and XCT expression in the young. But compared to the old, we saw this really a lack of expression in CD44 variant 9 and XCT, showing that at baseline levels, these are people with no ulcers, by the way. These are uninjured stomach. So even at baseline level, the aged individual doesn't express variant 9 or XCT. I performed IHC on a gastric ulcer array, and I found, so these are individuals with gastric ulcers, and you can see in the brown, this is expression of the activated form of beta-catenin. You can see in the young individuals, we had this nice healthy expression of beta-catenin. However, in the aged individuals, we saw almost no expression of activated beta-catenin. Next what I did was I wanted to see, is there some sort of therapeutic approach that we can take to help the elderly with the resolution of these injuries. So I took aged mice, gave them their acetic acid injury, and then I treated them with a P38 inhibitor. So the idea being that if we can treat them with this P38 inhibitor, all of these downstream effects could be sort of negated. We wouldn't see that loss in metastasis. We wouldn't see that decrease in cell, or we wouldn't see a decrease in cell proliferation, and we would see more healing in these individuals. So this was really exciting because I performed this in so many mice. I gave dozens of mice ulcers, treated them with P38 inhibitor for 7 days, and daily, and we saw this loss in, we saw an increase in gastric ulcer repair in the P38 inhibitor treated mice. And similarly, we also saw a loss in that fibrotic, any type of fibrotic development that you normally see in aged individuals during repair. So that was really cool, which suggests that we could be using a P38 inhibitor as a possible therapeutic approach for the elderly suffering from chronic gastric ulcer disease. I also looked at proliferation in these. So I treated with the P38 inhibitor. We saw an increase in proliferation, and you can see that reflected here with the BRDU. This is the young and this is the vehicle and the P38 inhibitor treated group. And likewise, we also saw a nice increase in CD44 variant 9 expression, which might be another something for future work to look at. So the conclusions that you can draw from this is that XCT can bring cystine in to provide defense against reactive oxygen species, where aging or drug treatment with drugs such as sulfasalazine can inhibit this process, leading to a buildup of reactive oxygen species. The reactive oxygen species can increase caveolin, which can redirect beta-catenin away from the membrane, or away from the nucleus to the membrane, where it can have all of these downstream nasty effects that lead to decreased repair capacity in the stomach. And similarly, SPEM is a reparative cell lineage expressed during injury. It expresses CD44 variant 9 as a robust marker for SPEM. And it also protects the stomach against reactive oxygen species by stabilizing XCT. And importantly, induction of beta-catenin is important for repair. And in the instance when CD44 variant 9 is lost, or XCT is inhibited, such as aging or drug treatment, you see a loss in repair, which could be rescued by P38 inhibitor treatment. So this is just one of my few projects. I have a few other ones. I'm looking at macrophage recruitment at the gastric ulcer site, and how macrophages may be secreting Indian hedgehog to initiate the CD44 variant 9 and XCT repair mechanism. And then another interesting project that I'm working on is I'm looking at circadian rhythm disruption after Helicobacter pylori infection, and how this may lead to a susceptibility in gastric cancer development in the elderly. So that's also a really cool project, and we'll be hoping to submit that one for publication soon. So if anyone wants to talk with me about that one afterwards, I have a PowerPoint for that too if you'd like to see some of the really cool data we have. So that's me, Nina, and Lauren, and Jayati, the members in my lab. So I'd like to thank them. I'd also like to thank all of the other past members in the lab who really contributed to the foundation of everything that I presented today, because without them, this wouldn't be my thesis project. All of our collaborators at Cincinnati Children's, our other collaborators that are not at Cincinnati Children's, and then also the American Physiological Society, University of Cincinnati, and Dr. Love for inviting me today. So.
Thank you for this terrific seminar on time.
Yeah, first of all, very nice seminar. I had a methodologic question and then more of a research question. The methodologic question, when you take the stomach or put it out, how are you able, I can't figure out in my mind's eye how you would take the stomach out and then induce an ulcer on the inside.
That's a good question. So so this method has been used for decades to induce a gastric ulcer. Previously, before that, and some people still use this other method today, the other method that people have used to study gastric ulcer, though, will give this chemical-induced ulcer via gavage. So but the problem with this is that it's like a global injury, right?
So you actually put the acetic acid on the outside of the stomach?
Yep, it actually goes through to the other side, yeah.
Now my question has to do with your really nice data with the P38 inhibitor. You did that on aging mice. Have you done that on younger mice, which would certainly save your lab money in terms of keeping the mice, and saving them even more money doing them on your in vitro assay?
That's a good question. So there's a lot of data that I didn't show you here. We have a ton of data for this project. Some of the other data that I've done is I did a lentiviral transfection of our in vitro cultures. And I've treated them with sulfasalazine glutathione and P38 inhibitor, and we do see an increase in repair in the in vitro cultures with and without the lentiviral. It's an XCT lentivirus. So.
But just the scratch and then adding the P38 inhibitor.
Yeah, we did that too. Yeah, and we saw, yeah, it does. But we haven't done the young mice with a P38 inhibitor, but that's definitely something we could try to see if we could just kind of speed it up, you know. That would be cool. That's a good question.
Roma?
I'm interested very much. Now, you mentioned that 500,000 of Americans develop ulcer a year. So, have you read or thought about the aging population in other cultures? For example, other ethnicities where the diet is different?
Yeah.
What is it now?
We haven't looked at other cultures. That's a great question. We take what we can get in terms of the human tissue. We take what we can get from these surgeons, and they're anonymous, so I don't know what cultures they are. But that is a good question because really, while the American citizens do suffer from gastric ulcers, and predominantly the age population, what the society, so Asian cultures actually suffer tremendous amount of, there's something about the Asian population, I think it's something genetic, that they do suffer from a lot of gastric ulcer disease and really just GI diseases in general. So that would be a good direction to look at the Asian population.
Because in the Okinawa Island, the Japanese island, there are people that live in the 100s, way above 100, and they have practically no diseases.
Yeah, I've heard about that.
You know about this population. So aging is a very good, they are very good for aging stuff.
That's a good point.
And they don't seem to have any of this type of
Yeah,
problems.
yeah. No, that's a
So I
good
think
point.
the diet is extremely important.
Yeah, yeah.
Because in the Asian population in general, they use a lot of spices, they use a lot of things that are carcinogenic themselves.
Yeah.
So that's why
Yeah.
stomach cancer is very rare.
Yeah, that has been the point of speculation, is their diet, yeah, as to why they have such a tremendous increase in GI diseases over there. But we just don't see it as much in the United States. Yeah. Yeah.
How do the cyclooxygenases figure into all this?
We haven't looked at that.
Is there?
Yeah.
Who was that? Please go ahead and ask questions.
Did you? Sorry,
Reporting. Reporting sovereignty.
we'll meet later. I'm going to ask you about the circadian rhythm later, but.
Oh, yeah.
Did you see any inflammatory responses?
So my other project that I'm looking at is alternatively activated macrophages and their infiltration in the granulation tissue. We do see a lot of inflammation in these mice with the gastric ulcers. What's interesting with that, though, I'm interested in an Indian hedgehog. My PI is all of her work, she's a hedgehog expert. But we do see a lot of alternatively activated macrophages at the site. And I've done work looking at, I've performed ELISAs with these macrophages and found increases in Indian hedgehog secretion after about 3 days. So.
Are you referring to M2?
Yeah, M2 macrophages. Yeah. I think you had a question. I didn't hear that.
Later.
Yeah, you asked a question.
Well, it was a general question as to how the cyclooxygenases tie into this.
Okay. We haven't looked at the cyclooxygenases. Yeah.
questions for the
I
student?
was waiting for the question of Dr. Solenti about the circadian rhythm, but then she's going to ask you later. So I'm asking you now. The circadian rhythm is associated, apparently also changes frequently with breast cancer.
Okay.
And I'm wondering if you have read or know about other types of pathologies that develop because of that factor. And so, what would be what is it must be a common factor that is inducing all of these cancers.
Yeah.
What do you have speculated?
Yeah, so I'll show you the mechanism that we proposed. I have a nice slide that I can show you. But we do see circadian rhythms, especially even with cell cycle. Cell cycle is circadian. It's governed by circadian rhythms in all sorts of different tissue that's been shown in research. What's interesting with what we're looking at with the stomach is we can infect our gastric organoids with Helicobacter. I can just take a needle, a very small needle, and just inject it to the inside of the organoid. And what we can see is disruption of cell cycle. If we just do Brucella broth, which is our control, we see this nice circadian rhythmic expression of cell cycle that is just completely demolished with Helicobacter infection. Similarly with aging, we looked at aged organoids. We can see not a disruption in cell cycle, a circadian cell cycle expression, but we see a dampening of it. So it's like the circadian rhythms in the aged population are just not robust at all.
Are you saying that you can still see the circadian rhythms in organoid cultures?
Yes. So a lot of people, when they think of circadian rhythms, they think of light, they think of sleep, right? But there's also an endogenous circadian rhythm that's still being kind of going on by itself in the cells without that control. So, but we certainly do see it in vitro. Yeah.
Students in the back.
I'll ask you later because I'm new to you.
Okay.
But I have a question. What's you use the reagent, the inhibitor SSD, why want to suppress the one included?
I'm sorry? Why?
That's good reagent to test the inhibitor that transporter. So what's the reagent used for pharmacology?
Sulfasalazine.
Yeah.
It's sulfasalazine. Sorry, I don't. What is the question? Sorry.
It's good to have a reagent that you can use an inhibitor as a tool for your experimental study. Is it clinical use drug or just?
Yeah. They treat it they use it for IBS, and some other things. I'm pretty sure for cancer as well. No.
It could be used for irritable bowel syndrome, inflammatory bowel disorders. As a dermatologist, we use it for neutrophilic types of dermatosis, similar to dapsone.
Yeah.
Okay, following asking a question. So we just, I'll just discuss, Richard discussed the paper in the lab, this new term called pyrotosis. If you use drug means inhibit the antipode or cystine, then you should generate so-called pyrotosis. It's generally the cell death, more or less like programmed cell death, but non-apoptotic pathway we call pyrotosis. The main reason for the cell death or cell suppression is generally the numbers, stress, or mutual loss.
Yeah.
We have that kind of interesting people think about the other genes involved in this pathway can dive into the diet.
Yeah, that's a good point.
Any other questions? Well, I just wanted to tell you that Vanderbilt or Harvard. And that's the next step. I wish you the best in your career. Thank you for